This video demystifies CRISPR gene editing, what CRISPR-Cas systems are, how guide RNAs and Cas9 cut DNA, and why that enables therapies, crop improvements, and ethical debates. You'll learn mechanisms, applications, and limitations. Chapter 1: Origins and Biological Foundation. CRISPR systems originated as adaptive immune defenses in bacteria and archaea. In 1987, researchers observed clustered, regularly interspaced, short palindromic repeats. The term CRISPR was coined in 2002. Bacteria capture short spacer sequences from invading phages and integrate them between repeats. These spacers are transcribed into CRISPR RNAs, cRNAs, that with Cas proteins recognize complementary viral DNA. The most widely used CRISPR tool is Cas9 from Streptococcus pyogenes, a roughly 1,368 amino acid endonuclease guided by a 20-nucleotide RNA to a DNA target adjacent to a PAM sequence, NGG. Understanding this natural mechanism enabled repurposing CRISPR as a programmable genome editor. This repurposing began in 2012 when researchers showed Cas9 could be programmed with a single guide RNA in vitro and in cells. Chapter 2: The Mechanism: How CRISPR Makes Edits. At its core, CRISPR gene editing is two parts: a programmable guide RNA and a nuclease, a single guide RNA, sgRNA, of about 20 bases pairs to a complementary genomic sequence next to a short PAM motif. For Cas9, NGG. Cas9 induces a double-strand break, usually 3 nucleotides upstream of the PAM. Cells repair that break via non-homologous end joining, NHEJ, which often creates small insertions or deletions, indels, or via homology-directed repair, HDR, if a donor template is available. NHEJ is efficient and error-prone. HDR enables precise changes, but is typically much less efficient in somatic cells, often less than 10% without optimization. Chapter 3: Real-world Applications and Examples. CRISPR has rapidly moved from lab to clinic, agriculture, and diagnostics. Clinically, ex vivo editing of hematopoietic stem cells using CRISPR-based edits, example, CTX001, exa-cel, has produced transfusion independence in patients with beta thalassemia and symptom relief in sickle cell disease in early trials. In agriculture, CRISPR-edited crops, for example, a non-browning mushroom with a disrupted PPO gene, have been developed without transgenic DNA. In basic research, genome-wide CRISPR knockout libraries contain tens of thousands of sgRNAs to identify gene functions and drug targets. CRISPR-based diagnostics, Cas12, CTX13, can rapidly detect viral RNA or DNA, enabling point-of-care assays. These applications show broad utility, but each carries technical and regulatory challenges. Chapter 4: Challenges, Next-Generation Editors, and Ethics. The main scientific challenges are delivery, specificity, and ethical limits. Delivery methods include viral vectors, AAV, with a 4.7 kilobase packaging limit, lipid nanoparticles, electroporation, and ribonucleoprotein, RNP complexes. RNP delivery often lowers off-target effects because Cas9 is transient. Off-target cleavage led to development of high-fidelity variants, eSpCas9, SpCas9HF1, and computational guide design to minimize unintended cuts. Next-generation editors include base editors, cytosine to thymine or adenine to guanine conversions without double-strand breaks, and prime editors, 2019, capable of diverse, precise edits. Modern methods can reduce detectable off-target events to very low levels in many experiments. Ethical and policy issues, highlighted by the 2018 germline editing controversy, require robust regulation, transparent trials, and public engagement before germline applications proceed. CRISPR transformed a bacterial immune system into a programmable genome toolkit. Guide RNAs direct Cas nucleases to cut DNA, producing edits via cellular repair. Its powerful medical, agricultural, and diagnostic uses are tempered by delivery, specificity, and ethical challenges, prompting safer editors, rigorous testing, and public policy.